Today I will talk about amino acids and their metabolism. Embrace yourself. This will be a long presentation, and I encourage you to stop after certain sections and review the material. So the first section, short section, I will talk about the structure and the sources of amino acids that we need for building our body and producing important bioactive products. In the second section, I will talk about amino acid metabolism. And in the third section, I will focus more on urea cycle, the detoxification system in the liver to remove ammonia, which can be liberated from amino acids. Again, the Word file is in the basic metabolic processes folder on Entrada. You can read it. And I found a very short overview of the urea cycle for you on YouTube that you may want to watch. So let us start with, here is again the intermediary metabolic process. You remember that everything which is set in blue is carbohydrate metabolism, set in yellow is lipid, fatty acid, triglyceride metabolism, and then here are the green stuff which are the amino acids. So this is what we will cover today, including the very important cycle in the liver which always accompanies amino acid degradation, which we call urea cycle. Of course, to take ammonia liberated from the amino group of the amino acids and produce urea, which can be freely excreted through the kidney. Sources of amino acids and the basic structure. Now, of course, everybody knows that the amino acids contain an amino group and a carboxyl group and a so-called side chain or side group bound to the alpha carbon. The chemical nature of the amino acid is determined by the nature of the side group. So there are acidic amino acids, for example, glutamic acid, or aspartic acid with negatively charged groups on the side chain, positively charged like lysine, neutral amino acids, and there are also additional sulfur-containing or aromatic ring-containing amino acids used in a body. Important to remember that we have 20 different amino acids that we can build into our proteins when the proteins are synthesized by translation of the signal from the different genes of proteins. Then the machinery brings in amino acids and puts together the protein. However, we have much more variety of amino acids that are built into our proteins because when the nascent protein is coming off the ribosome, right away post-translational modifications can be made. So the variety of the amino acids in our proteins are not just these 20, but also the modified versions. Let me introduce the categories of the amino acids. You probably already know that, but let me remind you that we have non-essential and essential amino acids. Essential means that we cannot synthesize them, so we have to take them up with food. We have then glucogenic, ketogenic, and glucoketogenic amino acids, which actually tells you the fate of the amino acid, which means that the carbon chain of the amino acids of glucogenic amino acids can be used to produce glucose in the liver, ketogenic again in the liver to produce ketone bodies, and glucoketogenic means then one part of the side chain of the R chain of the amino acids can be used for glucose production, part can be used for ketone production. I will go into details a little bit later. So where do we get these amino acids? Of course, the major source is dietary proteins, We need to take up dietary proteins. We need to take non-essential amino acids into our body. However, and I cannot emphasize it enough, our proteins are in constant turnover, which means that degradation of our own proteins also supply amino acids, and these amino acids will be either utilized by the body or will be eliminated. We can synthesize non-essential, we cannot synthesize essential amino acids. So what do we use the amino acids for? Um, some of this might be a surprise to you because there is a vast amount of molecules that we can synthesize from amino acid uh, carbon chains or even utilizing the amino group for that. So you will learn about synthesizing these very important molecules in this lecture and later on in different courses. But let me just start that amino acid pool is of course used every second of the life of the cell to replenish the proteins that the cell needs, either intracellularly or extracellularly. Now, these are the so-called bioactive products. First of all, glucose, second of all, ketone bodies or fatty acids. But please be aware that when we go further in metabolism, you will see that we cannot use the amino acids all the time to synthesize glucose or to synthesize ketone bodies or to synthesize other energy carrying molecules. It all depends on hormonal signal on the liver, which way the amino acids will be used. The other bioactive molecules, we will talk about a couple of them today, but we will also revisit later the purine and pyrimidine synthetic pathways, which heavily use amino acids to form our nucleotides. All right, the metabolism inside of the cell or taken proteins from the outside for recycling to amino acids is a very important feature. I am not going into great details here, but you need to remember again that our pool of protein is in constant turnover. We do have some proteins that the half-life is very long, like our collagens, especially in the cartilage, for example. The half-life of the collagen in our cartilage is as long as we live. So there will be some collagen molecules that we were born with that we still have later. But we need to recycle molecules, proteins. Proteins can be degraded intracellularly or extracellularly, can be damaged, can be modified, and the cell needs to determine which protein needs to be eliminated. So there are two pathways. One is intracellular proteins that are deemed to be non-functional within the cell. There is a system which we call ubiquitination, which ubiquitin will bound covalently to the targeted protein, cellular proteins, and it brings the protein into a proteasome, which will cleave the protein into amino acids. Now, when we have extracellular proteins, and some of the intracellular ones can go to the lysosome. The lysosome has an acidic pH. There are very specific proteins sit in the lysosome, and this is the recycling center of the body. It cannot just recycle proteins to amino acids, but it can degrade every other kind of molecules and releases building blocks that the cells can reutilize. So we have the amino acids. Amino acids are coming from the diet and or spit out by the cells and going to another cell, and they need to be transported into the cells for further utilization. We have active transport systems in every cell to take these amino acids and put them through the membrane. Now, active transport means that we need to use ATP for that transport system. And there are many different transporters. One is called COL, which transport cysteine, ornithine, arginine, and lysine into the cells. What are the clinical relevances of this active transport system? Of course, we know problems with the transporters, and it means that cells cannot take up, or certain cells cannot take up certain amino acids. But let me focus on one, which we call cystinuria, which is using this particular amino acid transport system. And it is not only in the surface of our cells in general, but this transporter is also on the tubular system of the kidney to reabsorb cysteine. Now, cysteine is the oxidized form of cysteine, which is the amino acid, and if it is not resorbed by the kidney, then cysteine will precipitate and form kidney stones. So this disorder is called cystinuria. There is also another disorders that I wanted to mention and related to another transporter, which is a defect in the intestinal absorption and the renal reabsorption of neutral amino acids, including tryptophan. Why I am emphasizing tryptophan? Because tryptophan is the starting material to produce a lot of bioactive products, which you will see later: serotonin, melatonin, and niacin, which is a vitamin, vitamin B3, NAD is the active form, will not be produced, and that has dire consequences. So another transport system, which is called gamma glutamate transport system, is actually using an amino acid, glutamic acid. And amino acids bind to the glutamic acid, and they are transported to the cells. Now, what is specific about it? That why we call it gamma glutamate, that glutamic acid is binding these other amino acids not through the alpha amino acid, but through the gamma carboxyl group of glutamic acid. So the carboxyl group, which is at the end of the side chain. To bind an amino acid to the gamma carboxyl group of glutamate, we need a specific enzyme, which is called gamma glutamyl transferase. It is especially pronounced or present in the liver. So actually GGT is used to detect liver damage. When liver cells are damaged, this intracellular enzyme is spit out, and you can detect the GGT level there. Gamma carboxyl glutamate is actually a glutamic acid, but you also saw this gamma carboxyl linkage to another amino acid in glutathione. If you remember, gamma glutamyl cysteinyl glycine is glutathione. All right, you can take a break now and review the material, but I will go further to amino acid metabolism now. Amino acid metabolism, I will talk about 4 different aspects of it. At first, I will talk about the removal of amino group, which is actually important in catabolism of amino acid, but you will see that transferring an amino group from one amino acid to a keto acid will produce another amino acid. So it is also important in synthesis of amino acids. And I will talk about also the bioactive products that are produced from different amino acids. This is a lengthy section. So again, you can stop at any point and you can review the material, or you can go through the whole thing and you can stop there before I switch to the urea cycle section to review the material. Just a reminder again, because it is important for understanding in metabolism, that we have essential and non-essential amino acids. We can catabolize all, but we cannot synthesize these. We can only synthesize non-essential amino acids. And you will see in the catabolism of amino acid section that I will talk about how amino acid, glucogenic and mixed function amino acid carbon chains can be used for glucose synthesis, and which are these two only, the only ketogenic amino acids that will be used for ketone body synthesis. So let us talk about removal of nitrogen from amino acids. That is very important for many reasons, as you see. So it is important when we break down amino acids and we want to utilize the carbon chain. When we want to utilize the carbon chain for energy production, or we want to utilize the carbon chain to synthesize other molecules, we need to remove at first the amino group, and we need to handle this amino group or ammonia so that not to poison ourselves. Now, we also use this purpose, the removal of nitrogen, when we want to incorporate this nitrogen into other compounds. So not excreting it, but transferring it to something and producing nitrogen-containing molecules. And this is very important note again, that we need to remove amino groups from amino acids, but we need to deal with that amino group, and we need to eliminate ammonia at the end from our body. So alpha amino acid, until the alpha amino group is on the amino acid, it is not channeled to breakdown. So we have to remove the amino acids. When it is removed, we can use the carbon chain to do whatever we wish to do with it. So let me remind you that there are these amino acids here, and what you see to be converted to pyruvate or oxaloacetate, or part of the chain of these amino acids to fumarate or succinyl coenzyme A or alpha ketoglutarate, so either to pyruvate or part intermediates of the Krebs cycle, we can use them to produce glucose. So in the liver, these amino acid carbon chains will be used or could be used for glucose synthesis. However, there are amino acids, and especially please remember leucine and lysine, which carbon chain can be turned into acetoacetate, then can be turned into acetyl coenzyme A. This can be used under certain hormonal conditions to produce fatty acids, and many times can be used to produce ketone bodies. Now, ketone bodies are very important for some cells to actually switch from glucose utilization to an energy-carrying molecule, lipid-based energy-carrying molecule, which we call ketone bodies. So why did I say leucine and lysine to remember? Because the carbon skeleton of leucine and lysine cannot be used in any way to glucose synthesis. It can be just used to ketone body synthesis. Phenylalanine, tyrosine, tryptophan, you saw them elsewhere, can be used either to produce ketone or produce glucose. I put here something important. When we take out the amino groups of these amino acids, they are channeled to the urea cycle for elimination of ammonia to urea. So, two steps are involved in removing nitrogen from an amino acid. One is called transamination, the other is called oxidative deamination. And I put here a summary figure just to show that if there is an amino acid, the amino group can be removed and then the carbon skeleton remains. Through transamination, the amino group is transferred to another keto group and another amino acid will be formed. I go into details just in a second. In oxidative deamination, we also produce a keto acid, but we remove the ammonia, the amino group in the form of ammonia. We are not passing it to another keto acid. So this is the final removal of the ammonia, which needs to be channeled to the urea cycle in the liver to produce urea. Now let's see these two different processes. Transaminases, also called aminotransferases, catalyze the transfer of the amino group of one amino acid to a carbon skeleton and produce an other amino group. Simple enough. We have two aminotransferases that are especially important. Why? Because they take the amino group from, after channeling from amino acids, they take them to alanine or take them to aspartate. Now, this is alanine aminotransferase or ALT, and this was in olden time called GPT. So what does it mean, ALT? Alanine is giving an amino acid to produce glutamate, putting the alanine amino group to alpha ketoglutarate, produce glutamate and pyruvate. You remember alanine is the major source for gluconeogenesis. Pyruvate is channeled into the gluconeogenic process. Now, you produce glutamate. Glutamate can be used by AST or aspartate aminotransferase, and now it is named from glutamic acid, giving the amino group to oxaloacetate and producing aspartate. Please remember that both of them are working with vitamin B6, which unfortunately I didn't put here in words. So vitamin is very important to work with these aminotransferases, and both of them can work in either direction. So it's reversible. So they can support amino acid synthesis and they can support amino acid degradation process. Let me put here something that you already learned about, and this is the alanine glucose cycle. A couple of things to remind you. So here is the skeletal muscle, and you may recall that skeletal muscle can use amino acids to channel the skeleton of the amino acids to alanine. Alanine going out and going to the liver, going to the circulation, going to the liver, and then it can be used for gluconeogenesis. This figure doesn't show you that because it follows the ammonia. Okay, so this is very important. This is the glucose-alanine cycle. Now, but more is happening in the skeletal muscle. Protein can be broken down not just to supply alanine to go into the liver for gluconeogenesis, but also it can supply energy for the muscle itself. And especially the branched chain amino acids are used to produce energy. In the meantime, they need to channel the amino group to an amino acid, which will be glutamate. So these are transaminases. Glutamate can be channeled to alanine to go to the liver, or glutamate can be further aminated to produce glutamine. So two amino groups are handled there, and glutamine can go either to the liver or it can go directly to the kidney for elimination. Now, following the nitrogen, when alanine goes to the liver, the amino group is transferred to glutamic acid or glutamate. Glutamine can be deaminated and serves as a nitrogen for urea, and urea will be eliminated. Okay, so very complex but very nice system. Transaminases working there, deaminase is working there, and glutamine synthase and glutaminase is working there in the whole cycle. Now let's go back and talk about the clinical relevance of these transaminases. So transaminases can be found in a lot of cells. Very important to focus on these two transaminases because they can be released from the cell to the blood. They can be detected in the blood, and that is indicative of liver disease. So AST, you remember, and ALT, both of them are working in the liver. They are intracellular, so the blood level of these transaminases are low. When tissue damage happens, when cellular damage happens, they are released to the circulation. This is an example of how ALT, one of the aminotransferases level, increases after ingestion of different poisons, for example, echinacea in the liver. It also can be released in heart attack. It also can be released with muscle damage, but it's important to remember that we do use it to look into liver damage by looking into the ratio of AST over ALT. So both of them will be increased, but if ALT ratio goes above 2, it is more indicative of liver disease. So the second system to eliminate ammonia from an amino acid is oxidative deamination. Why do we call it deamination? Because from an amino acid, ammonia will be produced and the keto acid will be produced of the carbon chain. Why it is oxidative? Because you see that when glutamate is, and this is glutamate dehydrogenase, the name of the enzyme, glutamate is turned to alpha ketoglutarate with losing the ammonia, the amino group, then NAD will be reduced to NADH. Now, time to time, this enzyme is reversible. We need to use the opposite pathway to synthesize glutamate. And you remember, for every biosynthetic reaction that use oxidoreduction, we use NADPH. So alpha ketoglutarate and glutamate is produced, NADPH is produced. Now, this is stimulated by low energy of the cell. NADH is produced. The reducing power goes to the mitochondria and produces energy. And this is stimulated by high energy level of the cell, which tells the cell that you can invest energy and produce glutamic acid, and from glutamic acid, other amino acids. All right? Now, it depends which direction we are going. It depends on cellular milieu. For example, if we are eating protein-rich meal, and the glutamate level is increased in the liver, it can easily push the glutamate to produce alpha ketoglutarate and produce energy through alpha ketoglutarate in the mitochondria, or through the reducing power of NADH going to the terminal oxidation system. Of course, you need to know that it's ammonia which is produced, and it needs to be eliminated through the urea cycle. All right, so I mentioned that there is a so-called glutamine cycle, which is also working, which takes glutamate and salvages an ammonia in the form of glutamine, or takes glutamine and through the glutaminase enzyme liberates ammonia. This is usually working in many cells, especially in liver, but also in the skeletal muscle, to eliminate ammonia, and glutamine can directly go to the kidney or to the liver and releases the ammonia there. If it is in the kidney, ammonia right away is eliminated in the form of ammonium ion. All right, this is a picture to put everything together. So when we want to dispose ammonia, then we have the amino groups of amino acids transferred through transaminases to different keto acids and produce amino acids that will end up in glutamate. Glutamate then can be deaminated and produce ammonia, which needs to go to the urea cycle. When we synthesize amino acids, these systems are working in the opposite direction. Ammonia and alpha-ketoglutarate will produce glutamate, and glutamate through transaminases and using other transaminases also can produce amino acids. Very smart system. Now, we are going further to look into how can we synthesize some amino acids. Again, just a reminder that we only can synthesize non-essential amino acids. Very quickly, non-essential amino acids can be synthesized when the carbon skeleton can be taken out from other processes. For example, serine can be synthesized from a glycolytic intermediate 3-phosphoglycerate. This serine can be used further modified to glycine, further modified to produce cysteine, the amino acid cysteine. Alanine also comes from pyruvate, which is an intermediate or end product of glycolysis when we talk about aerobic cells. So very smartly, we use molecules that are already available, the skeleton of the molecules, and we transfer amino groups to this carbon skeleton by transamination. There are amino acids that can be synthesized from Krebs cycle intermediates. So aspartate, asparagine from aspartate, glutamic acid from alpha-ketoglutarate, glutamine from glutamate. So oxaloacetate and alpha-ketoglutarate from the Krebs cycle can result in amino acid synthesis that they can further modify. All right, so degradation of individual amino acids now. Remember, we can do all. We remove the amino group, we now have the skeleton. So removal of the amino group is very important, which we went through. Then the carbon skeleton can be converted into intermediates, then can go to energy producing or metabolic pathways. And it can also be turned into intermediates that can be metabolized to further energy, carbon dioxide, water, or then they can go to glucose and ketone bodies. So the major carbon skeletons of amino acids that are degraded further: pyruvate, intermediate of tricarboxylic acid cycle, acetyl coenzyme A, and acetoacetate. So this is what shows you the glucogenic amino acids, which I showed you before. These guys here are glucogenic because they can be turned to Krebs cycle intermediates, oxaloacetate, which is now the starting point of producing glucose. When blood glucose level is low and glucagon and epinephrine is signaling to the liver, then amino acids will come from the skeletal muscle, as you remember, and will be used for glucose synthesis. When insulin is hitting the liver and we have amino acids after we have eaten coming to the liver, these amino acids can be used for energy. joining either tricarboxylic acid or turning to pyruvate going to the mitochondria. Right, ketogenic amino acids, as I told you before, they can yield ketone bodies. They can also use to produce acetyl coenzyme A. And when these amino acids, when the liver signals glucagon and epinephrine, they will stop at producing ketone bodies and release ketone bodies, liver cells. When insulin is signaling, then these can go to acetyl coenzyme A, and this can go to the fatty acid synthetic pathway. They can, of course, can be used from acetyl coenzyme A to produce energy during insulin signaling. Now, let me tell you what bioactive products can be generated from amino acids. So I put together a tiny little bed-looking table for you. So please bear with me. This is very good when you want to review the following lengthy material. So these are the amino acids here. In parentheses, I put there what bioactive product, if any, can be produced from these amino acids. And I put here the name of disorder that I mention when I go through this metabolism, special metabolism of amino acids. These are the disorders that you need to remember of. These are the bioactive products that you need to remember of for the exam and also for the step examination. So, leucine, isoleucine, valine. Although I put here a lot of words, but I want you to remember two things. These are the so-called branched chain amino acids, and as I mentioned before, these are very important to supply energy during normal conditions, especially for the skeletal muscle. Now, how to remember them? This is an acronym, I live, which leucine, E or I for isoleucine, and V for valine. Why this acronym is good? Because this is very important for amino acid utilization for energy. So when stress hormones are hitting the muscle, these particular amino acids will be used for energy and saving the muscle to be able to work. Now, what happens when an important enzyme is missing from this pathway? And the enzyme name is alpha-ketohydrogenase. When it is defective, the branched chain amino acids cannot be used for energy generation. So leucine, isoleucine, and valine will accumulate, will be excreted through the urine, and they will give a specific sweet smell to the urine. This is why we call it maple syrup urine disease. And you can read about this, that it is a pretty devastated condition for the baby. Maple, sweet smelling urine would be okay, but the accumulation of these amino acids lead to a problem with the neurologic system, brain damage, and failure to thrive for the baby. Methionine is very important to remember. It is a sulfur donor to produce sulfur-containing amino acids. Cysteine, methyl donor, so active methyl group can be added to other molecules and also is the source of creatine synthesis. I put here this figure, and you don't need to remember the whole pathway, but this is what is happening in some of our cells. Methionine is taking the adenosine group from ATP and produces S-adenosyl methionine. This is the active methyl group carrier. So in the next step, methyl group can be added to other molecules, methylated other proteins are important. Methylation of purines and pyrimidines are important, and so on. So the byproduct here is S-adenosyl homocysteine, which will lose the adenosine group, and homocysteine will be produced. I will talk about the problem that homocysteine is accumulating in a second. Please remember that for this cycle to work and not hurt the cells and tissues, this homocysteine right away needs to be eliminated. And with the help of vitamin B12 and folate, this particular homocysteine will go back to produce methionine. So if this cycle is working without a problem, we have active methyl group, and then we went back to methionine. Let me talk about what happens when homocysteine is accumulating. So when there is a deficiency in either folic acid or vitamin B12, we have homocysteine levels elevated, which can lead into stroke, heart disease, vascular disease. Of course, with vitamin B12 and folate are also very important, and in the lack of vitamin B12 and megaloblastic anemia and neurological problems will develop. Just a reminder. Right, so here is homocysteine. Research shows that it can promote blood clotting, it can promote vascular problems, and it can promote thus heart attack. Now, the research is not the strongest, but there are examples of this particular pathway. So in general, B12 folate acid level being appropriate in the cells is very important. Otherwise, stroke and cardiovascular problems and MI can happen. So this is what we were talking about: methionine, active methyl group, homocysteine. So homocysteine also can go in a different pathway with the help of serine, another amino acid. It can go to produce cysteine. So cysteine is one of our sulfur-containing amino acids, very important to produce other bioactive molecules, and just to be there as an amino acid. So what happens when this enzyme cystathionine beta synthase is missing? Then we are not producing cysteine. So when it is missing, will result in so-called homocystinuria, homocysteine accumulating, and it results in dislocation of the lens in the eye, also intellectual disability and problems with the musculoskeletal, the skeletal, and the neurological network. So cystinuria, homocystinuria is one of the disorders that I would like you to remember of. Okay, let's switch to phenylalanine, and you actually know this system already from the vital fluids block. So phenylalanine needs to be hydroxylated to tyrosine to produce tyrosine. So phenylalanine is hydroxylated to produce another amino acid, which is very important. This enzyme is phenylalanine hydroxylase. And if this enzyme has a mutation, genetic mutation, and tyrosine is not produced, phenylalanine is accumulating. Both will cause dire problems. Why is it a problem? Because tyrosine is further modified to produce bioactive products. So DOPA, and which can be turned into melanin in the skin, then further modified to dopamine, which is an important bioactive molecule, neurotransmitter, further modified to norepinephrine and epinephrine, which is a very important hormone. So when this particular enzyme is missing from the system, there will be a problem of synthesizing all these bioactive molecules, but also the problem will be of accumulating phenylalanine. What can we do if we don't have enough phenylalanine hydroxylase? If we have phenylalanine hydroxylase, phenylalanine deficiency, phenylalanine will be turned into different molecules, which we call phenylketones. So phenylpyruvate is one of them. And this will be leading into not producing enough tyrosine and other molecules, but also accumulation of these phenylketones, which will be detected in the urine. Urine will be actually turned dark. Why is it a problem? Because phenylalanine accumulation, phenylketonuria happens, which is damaging to the whole body, especially to the central nervous system. Now, what we can do is diet. Diet is leaving out phenylalanine from the diet. Just leaving it out is not enough. Medication can be used to capture more phenylalanine so that phenylketones will not be produced, so phenylketonuria will not develop. Please remember a fact that we put aspartame into many of our foods, including all the diet sodas, because aspartame is sweet. But aspartame can produce phenylalanine when it is metabolized. So if you look at diet soda cans, it says phenylketonurics. This drink contains phenylalanine. So not necessarily phenylalanine, but it will be metabolized to that. Okay, so as I mentioned before, tyrosine is produced from phenylalanine, and it will produce melanin in the skin, dopamine in the corpus striatum, and then epinephrine, the hormone, in the adrenal gland. Very important biomolecule. Disorders of metabolism, albinism, tyrosinemia can be produced if tyrosine aminotransferase is missing. So there is no way of going to dopamine. And this is a very rare instant when an animal is actually showing signs of albinism. All right, let's go further. Arginine is a conditionally essential amino acid. What does it mean? Then it can be synthesized from glutamate, but it is not sufficient for growth. So what do I mean can be synthesized? It actually synthesized as an intermediate of the urea cycle. So wait a couple of minutes and we will be there. Glutamine, you remember I mentioned it, very important amino acid, takes glutamate, puts amino group there, glutamine synthase, glutamine is produced, very abundant in the bone, muscle, blood, and also taken up through the blood-brain barrier and involved in neurotransmitter synthesis in the central nervous system. Histidine is a semi-essential, which means that we can produce a little bit, but when during growth, it's not enough, so we need to take up with food. Precursor of histamine, very important molecule in the immune reaction to support the allergic produced in the allergic reactions in general, but also in the accumulating in the central nervous system during migraine. Glycine is non-essential, very simple, amino group, carboxyl group, and alpha carbon and 2 protons. This is built into many amino acids, many proteins, but let me fast forward and say that when we are building our extracellular matrices, collagen is the most important protein there. In any extracellular matrix, you will see collagen, and 33% of the amino acids of a collagen molecule, independent of what kind of collagen, is glycine. So very important in building our extracellular matrices. Aspartate is important because it can be transaminated to oxaloacetate, which means that gluconeogenesis can go on. But aspartate is also one of the amino acids that must join the urea cycle to produce arginine, as you remembered before, and produce urea. Tryptophan. Tryptophan is actually very important in nucleotide synthesis and is important to nucleotide synthesis to produce nucleotide cofactors, including NADH, which is niacin vitamin B3. So what happens when we don't have enough tryptophan? It will result in lack of vitamin B3, NAD, and it will result in so-called pellagra, which means rough skin, and it will result in actually 3Ds, not just dermatitis, which is pellagra, I mean the sign of pellagra, but it leads to 3Ds, diarrhea, dermatitis, and dementia. Serotonin is one of the bioactive products. You don't need to remember the pathways, but please remember that serotonin is a neurotransmitter, so it needs to be synthesized, and we go one step further. Melatonin is synthesized from serotonin, so from tryptophan, and it is very important molecule to regulate our circadian rhythm. Inhibit the secretion of neurotransmitters, and it puts us to sleep. All right, so you learned about a lot of disorders of amino acid metabolism. So let me show you that in the newborn genetic screening, many of the amino acid metabolic disorder key enzymes show up. So PKU for phenylketonuria, maple serum urine disorder key enzyme, homocystinuria key enzyme, and tyrosinemia key enzymes are screened. Citrullinemia, arginosuccinate aciduria, argininemia are linked to amino acid metabolism. They are actually linked to urea cycle disorders. Now, very important to remember, of course, that newborn genetic screening is not only for amino acid related disorders, but for other disorders too, like hypothyroidism, hormone level, galactosemia is for carbohydrate turnover, galactose metabolism, sickle cell disease for hemoglobin point mutation, and so on. I put here the incidence level. You don't need to memorize this, but you can see that the most abundant or most well spread incidence, unfortunately, is PKU in amino acid disorders. What other molecules can be produced from amino acids? Creatine phosphate, creatinine. You may recall creatine is very important molecule which is produced from glycine and arginine, and a methionine. A methyl group is coming from methionine in the form of S-adenosyl methionine. And this is a very important energy carrying molecule because when it is phosphorylated, then it contains one high energy group which can be easily removed and produces ATP. Especially skeletal muscle system is important to contain creatine phosphate. Now, creatine is turned over and produces creatinine, I think, if I pronounce well, which is excreted through the kidney. So the serum level of creatinine is actually indicative of how the kidney can filter out creatinine. So kidney disease is expected when creatinine level is going up. Right. Now let's switch to the third section, which is focusing on When ammonia is liberated from amino nitrogen-containing molecules, the vast majority is, of course, amino acids. What can we do? How can we detoxify? We don't detoxify it through the urea cycle. Again, liver specific makes sense because liver can use these amino acids to produce glucose, to produce ketone bodies, in which process ammonia will be liberated in situ in the liver. So having the urea cycle there in the liver makes a lot of sense. We want to remove ammonia because ammonia is highly toxic. Every animal will eliminate ammonia. Some of them can eliminate it in ammonia or ammonium ion form. Lucky for them. Birds, for example, can eliminate ammonia in the form of uric acid. This is very acidic, so when a bird blesses you, you can expect that some of your clothing will be actually damaged by this blessing because the uric acid in the bird's feces is acidic. We do it further. We don't do uric acid, we do urea. Urea is actually almost neutral, so it's okay to handle, water soluble, easy to eliminate. Now, urea cycle is in the liver. It is just a reminder that most tissues actually put extra ammonia into glutamate to form glutamine. Glutamine goes to the liver. Glutaminase liberates ammonia, needs to go to urea cycle. Liver also has glutamate dehydrogenase, which liberates ammonia, needs to turn into urea. And all the transaminases will put the amino group finally to glutamate, which will liberate, from which the ammonia will be liberated. So whatever liver is doing with ammonia, ammonia can be produced in situ, which needs to go to the urea cycle. Now it just links everything to the glucosalanine cycle to produce glucose. During this one, ammonia, goes to urea cycle. Inborn errors of metabolism, also this big group also contains urea cycle disorders when ammonia is overproduced, leading to very big problems with brain, with the central nervous system. Hepatic encephalopathy will result in brain damage. Now, this is just exactly the same to show you that ammonia is going in the form of ammonia and joins the urea cycle. And we also put ammonia into, or nitrogen through, aspartate going into the urea cycle because we are actually eliminating two nitrogen groups, one from ammonia, one coming through aspartate. So let's see the details. All right, so this is the urea cycle. Actually, it works in this direction. So here is the ammonia coming from many sources. It needs to react with carbon dioxide, which comes from bicarbonate. The enzyme is called carbamoyl phosphate synthase 1, or CPS1. Why is it 1? Because you will see another one in a nucleotide synthesis. Okay, so CPS1, carbamoyl phosphate, is produced. You see here, these are mitochondrial processes, liver mitochondria. This carbamoyl phosphate will join the cycle. Similarly to acetyl coenzyme A joining the Krebs cycle, carbamoyl phosphate joins the cycle, which starts with ornithine and ends up with ornithine. So carbamoyl phosphate, ornithine, ornithine transcarbamylase. So, sorry. These are the 5 enzymes that you need to remember, okay? producing citrulline. Citrulline going out to the cytosol. Citrulline now merges with aspartate, the whole molecule. This is the second nitrogen which is brought in. First nitrogen from ammonia, second from aspartate. Now, this is arginosuccinate synthase, which will do this merging. We have the two nitrogen and the one carbon for the urea. Carbon came from carbon dioxide. Next step, we lyse this molecule to produce arginine and fumarate. This is arginosuccinate lyase. Fumarate can go back to produce malate, malate to produce oxaloacetate, oxaloacetate produces aspartate, so aspartate can join again to the urea cycle for the next round. We produced arginine. You remember, some cells use these enzymes to produce arginine, and when arginine is taken out from the cycle, urea is not produced. Now, how do we produce urea? Is taking arginine and cleaving off the urea portion by arginase to produce ornithine, which can go to the second round and the third and fourth round of the urea cycle. So this is how we produce urea. Urea is in the cytosol, can be excreted and secreted to the circulation, go to the kidney and excreted by the kidney. It's a very logical cycle. I am just sorry that you need to actually memorize the name of the enzymes and the intermediates, very important, ammonia, aspartate taken for nitrogen, carbon dioxide for carbon, and arginine is also produced in this pathway. Clinical relevance. Blood urea nitrogen is a measure of how the liver and kidney works. If kidney is not working, blood urea nitrogen will be high. If liver is not working, blood urea nitrogen will be even lower than the normal level because it will not be produced by the liver. However, in case of liver disease, ammonia level will shoot up, and this is the measure of liver disease. Okay, enzyme deficiencies. Any of these enzymes of the 5 can be missing from urea cycle. How to detect which enzyme is missing? Because all of these will result, missing any of the enzyme will result in high ammonia level. We need to look into the other intermediate which is accumulating. So for example, citrulline will be accumulating next to ammonia if arginosuccinate synthase is missing. Citrulline will be produced, but it stops there. Okay? If arginase is missing, we don't produce urea, but there will be accumulation of arginosuccinate or even citrulline, and so on. So look for what is accumulating, and then you can determine which enzyme is missing from the urea cycle. Why is it bad that urea cycle deficiency can lead to ammonia? Because as I said already many times, ammonia is very toxic. And you don't need to remember right now, but when you are going through our curriculum, you also see clinically that a lot of different organ systems, especially the central nervous system, will be affected by ammonia, by free ammonia. Very important, it can even lead to death. All right, so this is the end of this very long presentation. This is a table which, again, I suggest that you use when you want to make sense of this material, when you study the material, and when you review it. And let me finish with the summary, and majorly this is what you need to know from this presentation for sure. Essential and non-essential amino acids. The fact that you can use some for producing glucose, some which are just ketogenic, and this is leucine and lysine. And then there are amino acids that can be used for both glucose synthesis and ketone body synthesis. You need to remember how we mobilize the nitrogen, the amino group, from amino acids. We talked about aminotransferases, glutamate dehydrogenase, glutaminase, and glutamine synthase. We talked about in details of urea cycle that will finally detoxify ammonia. Please know the details of that. Now, you also need to remember the amino acids are precursors of important biomolecules, and the diseases that are related to amino acid problems. These are metabolic problems together with urea cycle disorder. This is a transport system problem, cystinuria and Hartnupp's disease. Okay, so these are the major points. Hope you will remember most of it. Take enough time to study this material. It's really a lot. Thank you.